Thank you for the introduction. It's a great experience to me to be here with you, and it's a great learning experience. I've learned quite a lot these last 2 days, and I'm taking that home. I hope I can build capacity in my own country with what I've learned here. Now, how does the thing work? So the topic is food safety and the composition. But there are elements that I think we should get into context that are equally important before we get to the composition. And that is the seed development, and specifically the plasticity of the genome. You have to think of that when you look at the rest of it. We've heard the last 2 days the phenotypic and agronomic assessment. Extremely important. You can't ignore that when you do the rest of it. So let's come to the food analysis, where we focus on today. The contents of this presentation is the importance of composition analysis of conventional crop plants, the development of the comparative approach for risk safety assessment of GM foods, and then some issues to ponder on. Just as an introduction, food security, and you that have experience of it in countries where we come from, is extremely important. The food scarcity and the need for food. The statistics tells us 9 million people by the middle of the 20th century, and to meet that, the food production must increase by 70 to 100% according to FAO. And crop genetic production is one of the agricultural inputs that's extremely important. Globally, biotech production is increasing, and it's the herbicide tolerance, insect resistance, and then specifically the traits that are stacked. So, by introduction, there are the single traits, the stacked traits, and the nutritional enhanced abiotic are grouped in separate groups. In which the comparative analysis we've been done so far with single traits, but in some countries it's also required for stacked. And in some countries the stacks is the new trait will be assessed thoroughly, but then nothing is required. In my country, for example, where GMOs are regulated, this whole stack as such is a GMO, so that will have to be assessed and compositional. What you'll notice, and we experience, that the metabolic pathways are getting much more emphasis and will be much more important in the future. So that's putting pressure on the regulators to be more informed and giving more training, build more capacity. Conventional food safety and nutritional landscape, historically, how that was developed by trial and error, by selection and preservation of plant variants with desirable traits, then human preferences, the taste and the color, and then the toxicants. As far as trait and color is concerned, we developed a sweet potato with high levels of beta-carotene, very fleshy and orange color, but unfortunately it tastes like pumpkin. It looks like pumpkin, it tastes like pumpkin. So the toxicants, they are always present in various concentrations. And in crop plants you find all these ones, toxic amino acids, lectins, proteinase inhibitors, antigenic proteins, the peanuts example, glucosinolates, alkaloids, solanine in potatoes, cyanogenic glycosides, fibrous polysaccharides and saponins, and condensed and tannins. And there you have an example of cassava that contains all these, but in various concentrations, so not all of them are that important. This is, for example, a very serious concern in cassava, and that is the presence of this toxic substance cyanide And apparently when the cassava is not close enough to the market, it deteriorates, and there seems to be a correlation between the deterioration, the stress effect on the production of cyanide. Cyanide is metabolized in a very small quantity to thiocyanate, and at the expense of methionine. But this is an example of trial and error over many, many ages. People don't underestimate traditional knowledge. They've developed cassava so that they can eat it and have it as one of the major, I think it's standing eighth in the world as in food production used in the world. So people use various methods to prepare it and reduce the levels of the cyanide. So why compositional analysis of conventional crops? There's the voluntary, the trade, environmental impact, biological variations as benchmark, regulatory in some countries, variety registration in our country, for example. There's a labeling requirements in the processed food and the food safety issues. So then also in research. As I said, with the sweet potato, we're doing still continuing on with research and looking at other elements in it and see how we can improve that, but that's like with conventional plant breeding. The analysis are the same according to the experts in nutrition, and there's a tendency to develop to use more and more information. So composition analysis for conventional food crops is still important. It will, and I think it will remain important, whether it be for nutritional assessment or for safety assessment. Now we get to the GMO crops, and there the OECD and other organizations, international, the FAO and WHO, have developed the comparative approach, which is, according to OECD, the most appropriate strategy for the safety and nutritional assessment of GM food. It's focused on determination of similarities and differences between GM food and conventional counterparts. It's not a safety assessment in itself, but a key step in the process of safety assessment. This is from the European Food Safety Authority, and putting the whole picture into context. Starting from the top, you can't see it very well, I think, but the history of the use of the parent crop, the donors, and all these steps into it, but then getting to the compositional analysis, the comparative analysis, of which the composition analysis, molecular, phenotypic, economic are key items. And this part forms the hazard part, identification part of the assessment. Remember, this is a safety assessment within a risk assessment, and we were talking risk. This is what we're aiming for, determining the risk. There's a difference between safety and risk. So then it goes to the hazard characterization by intended differences, looking at the intended differences, the new gene products, the DNA, the RNA, and whatever is necessary, and where there's compositional alterations, then the assessment that could go into a more detailed analysis. And yeah, the unintended effects, where there are differences between the phenotypic and the compositional analysis. So that could, in a safety assessment, for example, toxicology studies. Thereafter, exposure assessment, risk characterization, and then conclusion and safety. So your composition analysis is key, again, in this assessment. Different issues, the comparative approach, the comparator, the history of safe use, that is quite a point of discussion. The key compositional substances, substantial equivalence, unintended effects, and then the biochemical pathways and metabolism. So when the concept was developed, why it should be a comparative analysis, there were a number of workshops, and from that, it was decided that food, to do it according to toxicological assessment, is not really the best way of doing it. Because it's not a chemical. We do working with a chemical, and I work with chemicals and toxicology. There are specific principles that you've got to abide by. For example, you have a dose range, and the highest in that should show effect, but you can't have it with a food. So there are many complications if you don't have you try to do it according to a toxicological assessment. So the comparative thing is much more appropriate. Now, specifically the history of safe use, because that is part of your comparison. Firstly, what is safety? According to Walt, it is a judgment. It's value-laden. It is value-laden. It's understood within a context of a society, of a culture, of politics and economics. We often use the word safety rather very generally, but that is actually what it means. OSC tried to define it, but they say it's a reasonable certainty of no harm. Now, actually, what is harm? That is another question. How do you find harm? I think with the environmental risk assessment, they're also battling with this. You know, in the problem formulation approach that they're following, there's a specific step where you have to determine what do you see as harm, and that is the whole society actually should be involved in that. But risk, and there's always a risk, as you see in previous presentations. So history of safe use was defined, or they tried to find it, a number of scientists, Constable et al. History, what is history? What is safe? What is use? A very complex issue. And important part of it is the compositional side of it, for safety. Let's link it to the materials for analysis. The test, that is a critical composition and elements of the modified variety. OECD has identified them. The comparator, a non-variety with history of safe use, the word history of safe use. Near isogenic line grown under identical conditions. And then the references, as we discussed the last few days. And with the increased questions, new questions on the validity of the compositional consensus documents of the OECD, those are still the, let's call the elementary, before they were nutrition enhanced and we started comparing with that. Even if we compare with those are already on the market, GMOs, with changed traits, there are differences. So I think that's the point that we can discuss later today, is it's really, OECD shouldn't be updated and brought more into context. So the references, conventional varieties or hybrids that are grown commercially in geographies of the field trials. This is, for example, cassava again, where we decided on these specific elements that are important, characteristics. You'll see that specifically the high HCN and cyanogenic glycosides is a very important part. But these are the characteristic ones, the endpoints that we are focusing on. But should there be changes in future, we have to reconsider that. So this is a focused approach, a search-like approach. I think it's a Karl Popper, is a philosopher, a German philosopher, that developed this approach, a search-like and a bucket approach. This is a very search-like because we're doing a risk assessment. We want to focus with whether are the real risks. So issues in this assessment, this is substantial equivalency. Do we always understand it? Or is our knowledge broadening now? We're getting more and more information and starting to get more a feeling for what is substantial equivalence. The test, the comparator, the references. And now with all the natural variability, the genetics, the environmental, abiotic and biotic stress, these give us a much more better picture of what to understand by the substantial equivalence. An example I took from Professor Parrott. I changed the picture because I didn't like the picture he had it on. I've got a very nice picture of soybean on it. And it shows the variety, the influence on the environment and the influence of the genetics. That is for isoflavonoids in soybeans. I don't agree with him on he says normal and always safe. I think we're still arguing on that, whether the high concentrations are that safe or specifically what but we're exposed to that. So natural variability in conventional maize hybrids. This diagram, a graph, actually showing the different amino acids in 7 varieties, 6 locations in 1 year. The different, you can see there's a very, very big variability. This is specific from the ILSI database. The protein, it varies from about 6 to 15. That is, what is it? Percentage dry weight. South Africa's and Argentina is more or less here in the middle. So I think we're safe. I don't know how you interpret those at the ends and how nutritious that would be. But that is a comparison that is all about. So then we get to unintended effects and of conventional crops. There's not recorded very accurately, or shall I say, you don't have found much publications on it. And I think it could be because of the very, very skilled practices that are conducted with plant breeding, so that all the rubbish is picked out before it reached the point of food safety where the consumer is concerned. As far as GM crops are concerned, there are a few examples. One is the yellow rice, that there were changes. It was an unintended effect. So there are unintended effects, but I think due to the competence of the plant breeders, they're much less than there really would be a problem. So as far as the regulatory part of it, this increased safety assessment will be required. It means more and more information, and from the signals to the stacks and to the nutritional enhancement, and then specifically the abiotic stresses. Stacked beans, increase in it the last 2 years, 2010 to 2011, countries 11 to 12 doing it. Global GM stacks from 32 to 42 million hectares. Global presentation increased from 22 to 26. South Africa is a large number of them already stacked. Then the metabolic pathways and all these, that comes into question. I don't go into that. I just mentioned that the nutritional enhanced crop, the African Biofortified Sorghum Project, so a consortium of a number of research institutes, and conducted in a number of countries, where they're working on enhancement of certain elements, lysine, vitamin A, iron, and zinc. So there's quite a challenge when it comes to the composition analysis of this. The water efficiency made for Africa, this project is now running in Africa, and major challenges, I will think. Now let us get to the more in detail why we do compositional analysis. And one of the big reasons is because of possible unintended effects. And with more sophisticated methods like the omics, it will be possible to look at the metabolic pathways. there, Rick Graff compared the number of publications in 2011 comparing GMs and non-GM crop varieties with or without attentional metabolic changes. The number of crop plants was 10, the metabolic changes 25 and not metabolic. And here are some of the results. I think some of the authors are here in this audience. The natural variation explains most transcriptic changes among maize. Gene expression profiles comparable with non-GM. Microarray analysis revealed that plant mutagenesis may induce more scriptomic changes than transgene insertion. Transgenesis has less impact on the transcriptome of wheat, grain than breeding, conventional breeding. And then these techniques are not really perfect. It's non-target techniques, gathering a lot of information, and now you've got to sort it out and try to interpret it. So, as far as increased food safety are concerned, what is the way forward with our assessments? Where is the place for omics in this study? One tends to feel at this stage you're looking at the bucket and the searchlight approach, and with the uncertainty as a buzzword, what we do with it. Are we filling a bucket full of a lot of information and we don't know what to do with it? How are we going to assess it? How are we going to interpret it? Those are questions that we've got to answer. So, food safety development, the seed development is important, the economic phenotypic assessment, and then the food safety analysis, with all these included. There are apparently two schools or two different poles in the assessment when it comes to assessment of GMOs, and specifically where we look at more the concern for unintended effects. One group would say that if that is so serious, why don't we include also the conventional parts? Why don't they do that all that studies also for the conventional, in order to detect, if possible, detect unintended effects? Whereas another group would say, yeah, well, we should rather do use a risk hypothesis when we do it. And I think that those are issues that is important, that we've got to discuss and see what is the importance of the compositional analysis is. So the issues that we can ponder on is like, should we do an in-product assessment? Another issues are the ethical, the socioeconomic, the cost of compliance. That is putting it in the broad picture of the world need for more food. So with that, I think my time is up. And that is from our backyard plant breeding with caviar species.
This is Imdadul Haq from Bangladesh. I don't know whether I heard wrong, that you mean that unintended effect, for example, yellow rice. Did you mean golden rice?
Sorry, I don't follow me.
The unintended effect,
yes.
as for example, yellow rice. Did you mean golden rice or just any other?
That is what I come from the literature. Perhaps you have better information than I have. Somebody called it unintended effects, but there's a different pathway that was revealed during the process of it. But perhaps you can explain it better than I do.
No, if it is golden rice, it is not unintended. This is already meant for.
Okay.